Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the splicing apparatus. Okay, so before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and if you are the newcomer, so please you just, and this channel obviously is helpful to you, so you just share and subscribe this channel, and after subscribing you hit the bell icons for getting the notifications of my uploading videos. Okay, so let's continue. Splicing apparatus. The splicing apparatus is what? The central component of the splicing apparatus for GU. Okay, and the splicing process already I described. I have explained in my previous video, so you just check out that one. And here the splicing apparatus that is helpful or that is involved in the splicing process, and this apparatus containing these snRNAs U1, U2, U4, U5 and U6. These GUAG introns are the snRNAs, and these called U1, U2, U4, U5 and U6. These are very short RNA molecules and associate with the proteins. Okay, this U1, U2, U4, U5, U6, these have some proteins, and these proteins are called the small nuclear ribonucleoproteins or snRNPs. So what I am saying that this GUAG in region, that are the small snRNAs that containing these U1, U2, U4, U5, U6, and these are the very short RNA molecules, and these associate or these containing some proteins. These proteins are called the small nuclear ribonucleoproteins, or in the short form snRNPs. So you just ignore the noise please and adjust with me. Okay, that attach to the transcript and form a series of complexes, the last one of which is the spliceosome. So spliceosome is the not first product when these RNA molecules with containing some proteins, they are involved, and at last the product is formed by the reactions with these proteins, you can say, or the RNA molecules. Then the last product, this is called the spliceosome, is formed. Clear? Next, the process of assembly of snRNP and the protein factors. How this assembly of snRNP, that means the very protein, the small proteins, and the protein factors, how the assembly of these occur, these are described in the following points. Okay, so first point is that the complex, this is called the E complex, this initiates splicing activity. Okay, sorry for the noise that coming from the outside. Okay, so here the E complex initiates a splicing activity, and this complex comprises U1 snRNP. What is that? This E complex that containing one snRNP, that is the U1. And which binds to where? This binds with the 5' splice site. This binds with the 5' splice site. And how they can bind? They bind by RNA base pairing. Okay, RNA base pairing. And branch point binding is another protein, and the short form is that BBP protein. It binds at the branch point. Okay, it binds at the branch point. And the splicing factor, this is another factor, this is U2AF, this is the splicing factor, and this binds to the polypyrimidine and members of SR protein family. Okay, again I repeat how are the factors, or you can say how the factors bind where. Okay, so first E complex that containing U1 snRNP which binds to the 5 prime splice site. Clear? And how they can bind? By the RNA base pairing. The branch point binding protein, this is the BBP protein. This also binds to the branch point, and this is another protein involved in this process. And the factor, this is called the splicing factor U2AF. This binds with the polymylimidine and the members of the SR protein family. These are the members of the this binds with the SR protein family and polymylimidine. Okay, sorry, not polymylimidine, pyrimidine. Okay, sorry, polypyrimidine. And this, what are the SR proteins? These SR proteins name is coming, or it is given. SR protein name is given. Why? Due to the presence of an ARG-ACR rich regions. ARG-ACR rich regions. ARG is what? Okay, and I have already explained different names of the amino acids in my previous video where I explained about the translation. So you just check out that video where the short form. Here I just have written the amino acid in the short form, but there I explained the name of the amino acids. Okay. So this presence of where this ARG and ACR rich regions, that's why the name of the protein is SR protein. Clear? Now this SR protein interact with where? This interact with one another via their ARG ACR rich regions and also bind to the RNA binding of U1 SN-RNP to the 5 prime splice site. Okay, so first what is happening here? This SR protein has the important role in the formation of the spliceosome or splicing apparatus, you can say. These SR proteins actually they interact with each other, and the SR protein name is given. Why? Because they are found generally in the ARG and ACR rich region, and they interact with each other. How? By their ARG and ACR rich regions. And obviously with them, or they can also try to bind with another U1 SN-RNP. This is the SN-RNP is the protein. So U1 SN-RNP proteins, and where? At the 5 prime splice site. Okay, at the 5 prime splice site. So this is the first step, how the where the factors and the proteins first bind and the process becomes started. Okay. So now the E complex is converted to A complex. So these processes or these factors bind with different spaces, or you can say the different point. That's way the E complex converted to A complex. If these proteins and the factors cannot bind with each other, and E complex never converted to the A complex. Clear? Now, when U2 SN-RP binds to the branch site. Okay, here another thing is happening that you can learn here that about the U1 SN-RP. But only U1 SN-RP bind with this, not convert the E complex to the A complex until the U2 SN-RNP binds to the branch site. Clear? So two SN-RNPs are involved here. One is the U1 and second one is the U2. Second point is that the pre-spliceosome complex, that is the A complex, it is just the compound, you can say, or the complex that is not already formed the spliceosome. So that's why it is called the pre-spliceosome, or you can say that is the intermediate form of the spliceosome. So that's why it is called the pre-spliceosome. Now this pre-spliceosome complex, that is the A complex, that comprises of U2 SN-RNP and then attached to the branch site, and at this stage an association between the U1 SN-RNP and U2 SNRNP brings the 5' splice site into close proximity to the branch point. So what's it saying? That nothing else, that due to the binding of this U2 and UN SNRNP at the branch site, as a result, that U1 and U2 brings to the 5' site 5' splice site into close proximity. Okay, so what is saying that the pre-spliceosome complex comprises the commitment complex plus U2 SNRNP, the latter attached to the branch site, and at this stage an association between U1 SNRNP and U2 SNRNP brings the 5' splice site into close proximity to the branch point. That means as U1 and U2 bind with each other, that's why the 5' splice site and the branch point both are close together. Clear? Or they are coming too close to there. Third point says that the spliceosome is formed when U4 U6 RNP and U5 SNRNP that attach to the pre-spliceosome complex. So when that U4 U6 and U5, the two are bind with them, which to the U1 and U2, then the spliceosome formation becomes start. Clear? And the B1 complex that is formed when U5 and U4-6 SNRNPs binds to the A complex. So now A complex is converted to B1 complex when U5 and U4 U6 both bind with the A complex, and that way A complex is converted to the B1 complex. Clear? Now okay, this B1 complex that converted to the B2 complex. Okay, B1 complex now converted to the B2 complex when after U1 SNRNP is released. Clear? So when U1 that is released from the B1 complex, so then B2 complex is formed, and this releases of U1 allows U6 SNRNP to interact with the 5' splice site. That means as U1 that is released from the B2 complex, so when the B1 complex is converted to the B2 complex, then U6 SNRNP that has a chance to bind with this B2 complex to form it. Clear? And this reaction is triggered by the release of the U4. And obviously, when U6 try to bind with this, that means U4 must be released from that region. Clear? And this requires hydrolysis of ATP. That means energy is also required for this releasing of U4. When U4 dissociates from the U6, then U6 SNRNP can pair with the U2 SNRNP and to form the catalytic active site to form the C complex. Simple word, that is, as U4 when dissociates, then what happened? This U6 that bind with what? U2. U6 bind with U2 and to form what? To form the C complex, and it is the active site. And this result in additional interaction that bring the 3' splice site close to the 5' site and branch point. So already 5' splice site that in the close proximity to the branch point, but when the C complex become formed, then the 3' splice site and the 5' splice site, they become close towards themselves with the branch point. Clear? And all three positions, that means 3' splice site, 5' splice site and the branch point, they all three positions in the intron are now in proximity, they close to each other, and the two trans-esterifications occurs when they all close to each other, that means 3' splice site, 5' splice site and the branch point, then trans-esterification occurs. Two trans-esterification occurs. Okay, I have explained the trans-esterification or what happening in this trans-esterification, so it is already present in my channel, so you just check out that one. And in that way the spliciosome formation is completed. Okay, in that way the spliciosome formation is completed.